I'd like to start by announcing that WebEVTalk is now on Patreon. So what is Patreon? So it's basically a website that allows us to receive sponsorship and do it so openly. So as you know that we're starting about a year ago, and all the administration costs was actually privately funded by me. And we also have a lot of support in the form of like time, valuable time from the volunteers. And so coming to the second year, and we have much more talk plan, and we want to have a little bit of sort of stipend for the person who does the YouTube, and also covering the costs for the WebEVTalk, which is pretty much a database and Zoom account and that sort of thing that we're going to move into WebEVTalk Zoom account. So if you like this format and you like to support us, so you can find the link in our emails and social media, or you can go to Patreon and look for WebEVTalk. So we have like individual industry and university. The industry and university can put logos and we will acknowledge them. So we have our first patron, which is Horiba Scientific, and I'd like to thank Horiba for supporting us. And with that, I'll pass it to Alissa to start introducing Natali. Thank you, Alissa.
Great. So I'm delighted to introduce Dr. Natali Chanaday, who was at Vanderbilt University. She obtained her PhD in chemistry at the University of Cordoba, Argentina. She then moved to the United States to work with Dr. Ege Cavallali, who's in the Department of Pharmacology at Vanderbilt. Her postdoctoral work has expanded our knowledge of the molecular mechanisms underlying different modes of synaptic vesicle release and recycling. And she's got a great story, so really excited to hear about it.
Awesome. So thank you for the invitation. It's truly like a big pleasure to do this and to be able to share our work. Our lab, it's more like a molecular neuroscience, neuropharmacology lab. And this is actually our first exploration of the extracellular vesicles field. So I'm very curious to know what you think about our project. My name is Natali, as you know. I'm a postdoctoral scholar, and the title of the talk, as you can see here, is Interneuronal Exchange and Functional Integration of Synaptobrevin via Extracellular Vesicles. And I think that for this audience, I don't need to introduce extracellular vesicles, so I will just be very brief. As you all know, there are different types depending on the route of secretion, and they will have like different sizes, different components. Historically, when they were discovered, they were thought to be more like a mean of elimination of debris or waste materials. But we all know now that they are an important way of intercellular communication. There is a lot of work on, in particular, nucleic acids that they can transfer in diseases like cancer or neurodegeneration, but also during normal functioning of many cell types. Almost every single cell type that we know of, they secrete extracellular vesicles. Also, this is a very interesting way to secrete soluble proteins that do not have like a particular signal for secretion, and also to secrete membrane components like membrane proteins and lipids that cannot be secreted through other mechanisms. So actually, a lot of work in the field has done looking at the nucleic acids that extracellular vesicles can transport, but we were more curious about membrane components. So I hope I will show you today a little bit about transfer of membrane components. And coming to the, I told you we are neuroscientists, so coming to the nervous system, there is some more functional work studying the role of extracellular vesicles, as I said, for example, during neurodegeneration or during axonal regeneration in the peripheral nervous system. There is some work showing the role during development, but more like a mechanistic molecular perspective on how they work and how they can regulate neurotransmission. it's a big, like a big hole in the field. We still don't know a lot. So that was our question. Our question was like, okay, let's try to isolate extracellular vesicles from neurons and try to understand their function from a neuroscience perspective, neurotransmission, and see if we can gain any insight into the molecular mechanism underlying that function. So I said we want to study extracellular vesicles from neurons. So the first thing we did was to develop a culture system that is free of astrocytes. Usually neuron cultures are co-cultures of astrocytes and neurons. So we basically made these astrocyte-free cultures. If you compare the content of astrocytes is greatly reduced compared to normal cultures. So now that we have this, we can isolate extracellular vesicles that will be secreted mainly by neurons. And then we did a clearing procedure to eliminate different cell debris and different cellular components to finally get the supernatant that we use for extracellular vesicles isolation. And we test 3 different methods that are used in the field. Method 1 is an old historical and controversial ultracentrifugation method. And then method 2 and 3 are commercially available. In case of method 2 is size exclusion chromatography, and in method 3, it's a membrane affinity column. You can see down here that if we measure the size using nanosite, the extracellular vesicles that we obtain via the 3 methods have similar sizes. And we also corroborated using EM that they have similar sizes and morphology. So we wanted to further characterize these vesicles, and we did proteomics. And these are the results. We found, as you know, if you did proteomics, a lot of proteins, but important for extracellular vesicles, we found a big proportion, around half of the proteins that were classical or proteins that are usually found in extracellular vesicles. To give you some examples, markers like the tetraspanin C81 and CD9 were found, also flotilin, fibronectin, annexin, many others. A tetraspanin that was not found, and I lost here, I don't know why it's not here, is the tetraspanin CD63, and I'll come back to that later. And also we did not find like ER proteins like endoplasmin or GP96, also the cis-Golgi protein GM130, which means that we probably do not have much contamination from cell debris. We also did not detect any astrocytic marker. Again, pointing that these are neuronal. So I want to bring your attention to one of the tetraspanins that we detect in the proteomics, the protein CD81. We went and did some confocal colocalization analysis, and we found that CD81 was indeed expressed in neurons. This is an axon, and you can see that CD81 is present at the axon, and it actually colocalized with the presynaptic marker synapsin1, indicating that it's a presynaptic terminals. So we designed an shRNA to knock down CD81, and you can see the quantification here. And then we measure the effect on that on neurotransmission. What I'm showing you here, and I will show you throughout the talk the same, is miniature inhibitory postsynaptic currents. This is a measurement of spontaneous neurotransmission mediated by the neurotransmitter GABA. And when you do electrophysiology, it looks like this. There are like these small peaks. Each one of them comes from the fusion of one synaptic vesicle at one synapse. And this is what we call the spontaneous release of neurotransmitters. So you can see here the control, but now, for example, if we knock down CD81, and you can see quantification here, there is a decrease, sorry, my English, a decrease in the frequency of spontaneous neurotransmission. If we overexpress it, on the other hand, now there is an increase. So this indicates that CD81 at neurons can modulate inhibitory spontaneous neurotransmission. Actually, we also tested doing rescues with 2 mutant proteins that affect CD81 binding to cholesterol, and they cannot rescue. So this indicates that cholesterol binding by CD81, it's also important for its function. Something to note is that the amplitude of this spontaneous neurotransmission is not changed. From our perspective, this usually indicates that the effect or the modulation is purely presynaptic, because the increase in frequency means an increase in presynaptic fusion, presynaptic release of neurotransmitters, but no change in amplitude means probably there are no changes in postsynaptic receptors. So CD81 has some presynaptic modulatory role in the release of neurotransmitters. And it is present in hippocampal neurons, and it is secreted in extracellular vesicles. So our next question was, what do extracellular vesicles do to neurons? Do they have a similar role as CD81 or not? So something before I move on, I told you before that we did not detect the tetraspanin CD63 in our proteomics. Actually, if we repeat these experiments, knocking down or overexpressing CD63, we see no effect at all. So this is something that is specific for CD81 and not a property that's shared by CD63. So now the fun part, what do extracellular vesicles do? So what we did is we isolated extracellular vesicles, we added them to neurons. The final concentration that we used in the experiment was the same concentration that there usually is in a supernatant in a culture. Then we incubated for different time points. And again, we measure inhibitory spontaneous neurotransmission. So you see the same kind of recordings as before. And we tested extracellular vesicles isolated by the 3 methods that I mentioned before. And as you can see here, as time goes by, there is an increase in the frequency of spontaneous neurotransmission, and that is quantified here. The number of peaks per time increased. Again, there are no changes in amplitude, so this indicates that extracellular vesicles are increasing the presynaptic release of neurotransmitters. Something interesting is, if we now isolate extracellular vesicles from neurons that lack CD81, those extracellular vesicles have no impact on neurotransmission. So I didn't show you the data, but only inhibitory neurotransmission is modulated, excitatory neurotransmission is not changed. But both spontaneous and action potential dependent neurotransmissions are increased. I'm only showing you spontaneous for a matter of time. And as I said, this seems to be dependent on CD81. Just to compare, I told you from the beginning that we were interested in understanding extracellular vesicles that are secreted by neurons. So to compare, we isolated, actually, no, I have something before. I'll come back, sorry. So the question is now, are these vesicles secreted in an activity-dependent manner? So that is this experiment. So what we did is we incubated the same as before for different times with extracellular vesicles, but now in the presence of TTX, which is a drug that will inhibit action potential. So the network activity in the culture will be downregulated. And now we compare the effect of EVs in the presence or in the absence of a drug. And as you can see here, in both cases, there is an increase in spontaneous neurotransmission when you compare to control. This means that at least the effect of the extracellular vesicles is not activity dependent. We also isolated extracellular vesicles in the presence or absence of stimulation. In the short time scale of like 10-20 minutes, we saw the same levels of secretion. So at short time scales, there are no effects or activity does not impact on the secretion of these neuronal extracellular vesicles. I'm not showing you the data, but trust me. And then we asked if this was calcium dependent. So we pre-incubated the neurons with BAPTA-EM to chelate calcium, and now extracellular vesicles cannot modulate neurotransmission. So this increase in neurotransmissions that extracellular vesicles are causing is calcium-dependent type of neurotransmission. And now the question that I had before, all of this is using neuronal extracellular vesicles. So to compare, we isolated extracellular vesicles from culture astrocytes. I didn't say this before actually, but we collect extracellular vesicles from 72 hours, either astrocytes or neurons, and we use mature cultures. So for neurons, we use cultures that are 2 to 3 weeks old, when the neurons are mature and synapses are formed. And from an electrophysiological point of view, the network is mature too. So we collect extracellular vesicles for 72 hours for all the experiments. Coming back to this, now we isolated extracellular vesicles from astrocytes and we compare with neurons. And as you can see here, where the neuronal extracellular vesicles cause an increase in the frequency of spontaneous neurotransmission when you compare with the control. Now, astrocytic extracellular vesicles have no effect. So this acute effect on neurotransmission that we see seems to be exclusive for extracellular vesicles of neuronal origin. So going back to the proteomics and going back to how I introduced myself at the beginning, I said that we're more neuroscientists that have a molecular perspective of things. So our question was, what's the mechanism? How are extracellular vesicles regulating this form of neurotransmission? So we came back to the proteomics, and on top of the typical extracellular vesicle markers that I mentioned in the beginning, we found that they have a lot of neuronal markers, as you would expect from neuronal extracellular vesicles. For example, some of them are adhesion proteins that are involved in development, but many of them are actually synaptic vesicle proteins, and in particular snare proteins. And these proteins are the ones that mediate the fusion of the synaptic vesicles, and in that way they regulate the release of neurotransmitters. So we hypothesized that maybe these snares that are being transported via extracellular vesicles, the ones that are underlying the increase in neurotransmission. So before I go into the result, I want to give you a little bit more background. This is a synaptic vesicle protein. It has on the synaptic vesicle membrane, there is this snare called synaptobrevin 2 or VAM2, which is a majoritarian snare protein. And then at the plasma membrane, at the presynaptic terminal, there are two partners, syntaxin 1 and SNAP25. And this complex of VAM2 with SNAP25 and syntaxin 1 is the one that mediates the fusion of the synaptic vesicle. What happens is that there is a calcium sensor called synaptotambin 1 that interacts with these proteins. And now, when there is a calcium rise in the presynaptic terminal, the calcium will bind to synaptotambin 1, allowing change in conformation, fully zippering of the snare proteins. And now there is fusion and release of neurotransmitters. And VAM2 or synaptobrevin 2, this protein that I told you there is the majoritarian mediating this process, was also the majoritarian snare protein that we found in the extracellular vesicles in our proteomics. So we wanted to study the trafficking of this protein from the extracellular vesicles after it gets incorporated into the neurons. Where does it go after it gets incorporated into the neurons? So to do this, we isolated extracellular vesicles from neurons that overexpress synaptobrevin 2 fused to fluorine, which is a pH-sensitive derivative of GFP. These extracellular vesicles now will have synaptobrevin 2 fluorine, and now we will incubate wild-type neurons with these fluorescent extracellular vesicles. Wait 30 minutes and perform live imaging experiments. And fluorine is I told you, a pH-sensitive GFP that is on the luminal side of synaptobrevin. So when it's inside the synaptovesicle, basically, the fluorescence will be quenched. But when the synaptic vesicle fuses, now the pH will equilibrate with the extracellular pH, and there will be an increase in fluorescence. As you can see here, these tiny peaks. This means fusion. And we used ammonium to reveal all the molecules that were present. So to show you more examples, you can see here before and after stimulation, we did a train of action potentials to simulate neuronal electrical activity. And you can see an increase in frequency. In structures that by eye, they look like en passant axons. So this is probably presynaptic. And there are many examples here, recordings from individual presynaptic terminals. And again, you can see an increase and a decrease in fluorescence, which usually means fusion and endocytosis of the synaptobrevin 2 fluorine that is on the synaptic vesicles. And this is an average of many, many experiments showing the same. We went a step further and we did some cofocal colocalization to see if synaptobrevin 2 fluorine was really at presynaptic terminals. And yes, there is a lot in the cell body too, but it is present at these punctated structures and it colocalizes with the presynaptic marker synapsin 1. So this means that synaptobrevin 2 can be transported from the extracellular vesicles to, after incorporation by the target neuron, it can be transported to presynaptic terminals. And in there, it's probably going to synaptic vesicles and it's undergoing normal rounds of fusion and endocytosis. So we hypothesize that this is how they are increasing the release of neurotransmitters. So if that hypothesis is true, if this is a mechanism, this will mean that the extracellular vesicles should be able to rescue synaptobrevin 2 knockout neurons. So that we tested that next. We make cultures of synaptobrevin 2 knockout neurons. I told you before, synaptobrevin 2 is the main snare mediating the fusion of synaptic vesicles. So if you knock it out, spontaneous neurotransmission is greatly reduced. You can see here the number of peaks per time is greatly reduced. But now, if we incubate them with extracellular vesicles, there is an increase in the frequency over time, and that is quantified here. So our hypothesis was proved to be true. Extracellular vesicles can partially rescue spontaneous neurotransmission in synaptobrevin 2 knockout neurons. Again, there are no changes in amplitude, indicating that this is a presynaptic mechanism. And this not only depends on synaptobrevin 2, but it also depends on CD81. But again, for matter of time, I'm not showing you all the data. To show you with more proof that this truly depends on the presence of synaptobrevin 2 at the extracellular vesicles, we now isolated extracellular vesicles from synaptobrevin 2 knockout neurons. So those extracellular vesicles now do not have synaptobrevin 2, and now they cannot, if you compare here with the control or this with the control here, now they do not modulate neurotransmission. So it's the synaptobrevin 2 that's being transported via extracellular vesicles that is increasing the release of neurotransmitters. And this is a dot plot to show you that extracellular vesicles from synaptobrevin 2 knockout indeed do not have synaptobrevin 2. Doing a circle and linking to the first results that I showed you at the beginning, we also did dot plots on extracellular vesicles isolated from CD81 knockdown neurons, and we could not detect synaptobrevin 2 in those extracellular vesicles. This indicates that CD81 is probably involved in the recruitment of synaptobrevin 2 to the extracellular vesicles. We also measure synaptobrevin 2 on the extracellular vesicles isolated from astrocytes, and again, we could not detect synaptobrevin 2. So all of this together really points to a role of synaptobrevin 2 on this modulation of neurotransmission. And just to finalize the final piece of data that I will show you something very interesting. Until here, I always showed you a modulation of inhibitory neurotransmission, that is neurotransmission mediated by the neurotransmitter GABA. But on the synaptobrevin 2 knockout neurons, now if we incubate with extracellular vesicles, excitatory neurotransmission can also be increased or modulated. So this means that the effect of the extracellular vesicles on neurotransmission actually depends on the endogenous levels of synaptobrevin 2 at different types of synapses. So to close, no, I have one last piece of data. We did some controls. So to kind of put together everything until now, our hypothesis is, or the mechanism that we propose is like extracellular vesicles have this snare protein synaptobrevin 2. When synaptobrevin 2 gets incorporated into the target neuron, it goes to synaptic vesicles. And this extra copy number of this snare protein is what increases the propensity to fuse of the vesicle. So it will increase neurotransmission. So if you remember, when I showed you the mechanism of synaptic vesicle, I have twice the same slide, or is it, oh, I'm so sorry, I have twice the same slide. Okay, this is the slide that I have. I'm so sorry. If you remember the cartoon that I showed you at the beginning with the fusion machinery, I told you that synaptobrevin 2 was the snare protein at the membrane of the synaptic vesicle, and then SNAP25 and syntaxin 1 were the membrane proteins, the partners of synaptobrevin 2. In our proteomics, we found that synaptobrevin 2 was present in the neuronal extracellular vesicles, but actually SNAP25 and syntaxin 1 were not present. So if we think that the modulation of neurotransmission is due to the snare proteins that are transported via extracellular vesicles, then snare proteins that are not present in extracellular vesicles should not be involved. So we tested this next on SNAP25 knockout neurons. Again, SNAP25 knockout neurons have very reduced neurotransmission. And now if you incubate them with extracellular vesicles, nothing happens because extracellular vesicles do not have SNAP25. So extracellular vesicles cannot rescue SNAP25 phenotype. But this is not because they're not incorporated, because if we, again, we do confocal, we can see that they co-localize with the presynaptic marker synapsin 1 to similar levels to wild-type controls. So there's still incorporation of the components of the extracellular vesicles in these neurons. It's just that they have no impact on neurotransmission. And this also strengthens the idea that the incorporation is not activity dependent, because these neurons that have almost non-neurotransmission, they still can incorporate these extracellular vesicles. And as I showed you, we repeated the fluorine experiments that I showed you before. And again, there is no traffic of synaptobrevin to fluorine in these neurons, again, because they do not have SNAP25. So now I will wrap up the mechanism that I showed you today. And we studied extracellular vesicles secreted from cold or hippocampal neurons. We found that the secretion and the incorporation is not dependent of activity on short time scales of like 10, 20 minutes. We don't know about long term. The tetraspanin CD81 is important for the recruitment of synaptobrevin 2 to the extracellular vesicles. And after incorporation, synaptobrevin 2 can be trafficked and incorporated into functional synaptic vesicles, and it can undergo rounds of fusion and endocytosis. And this is extra copy number of synaptobrevin 2 that the vesicles are getting, where it's underlying the increase in the neurotransmitter release. I would like to thank my lab, mainly my PI, Ege Cavalli, because he's amazing. This work was done with Ale Vilcaez from Argentina, and we had funding from here, from the US and from Argentina. I also want to thank Vanderbilt University because it's a great place to work. And thank you all for the invitation and for listening to me. And now I can take questions.
Great. That's always fabulous to see.
Yeah, I just feel bad because I made some changes in the presentation last night and I clearly didn't save them. So I had things like repeated. I'm so sorry.
So, wow, there's an echo here. Do you guys hear an echo? Or is it just me? You hear the echo?
No.
Okay. Okay. Oh, you don't hear it. Okay. Well, it'll just bother me then. So I will kick off with a first question while we wait for people to raise their hands or put something in the chat box. I was wondering, Natali, in your add-back experiments that you did, how did you use equal number of extracellular vesicles, and did you figure out, like, how did you figure out what amount of extracellular vesicles to use?
You mean when we incubate for the experiments, basically, how much vesicles we're adding for all the experiments that we did, right?
Yeah, because you have a lot of very elegant comparisons, you know, between knockouts and controls. And I guess I'm wondering, did you use like equal number of protein or equal number of vesicle? And how did you figure out what was the appropriate dose for your system?
So we knew, we measured the concentration of extracellular vesicles on the supernatant on our like common neuron cultures. And what we use for the experiments is the same concentration. And we measure the concentration in number of particles per milliliter. And we use for all the different groups, we always use the same concentration in number of particles per milliliter.
I see. So you were trying to match what you saw in conditioned media, basically.
Yes.
Gotcha.
We tried, actually, I can show you the figure, but we tried using, we actually did a dose, we ended up using that, but we did a dose response curve using like less than what is found in the culture media, and double and 5 and 10 times more. And actually the effect in neurotransmission is, it has a dose response. It saturates when we start to use like around 4 or 5 times more than what is usually in the culture. It starts to saturate. And also for electrophysiological experiments, the baseline starts to be very noisy. And I think it's because we're adding a lot of lipids on the media. So the membrane starts to, you know, be very noisy. So we ended up using the same concentration that we have in the cultures. But actually, I can show you if I can share my screen again. It's this one here. You can see if we add more extracellular vesicles, the effect is larger, but it also appears earlier in time. So for when we use the same concentration as we use in the cultures, the peak in the effect appears after 30 minutes. But if we use more, that peak is higher and it appears at 10 minutes. So it's also, the effect is also faster.
Gotcha. Okay. I see Jason Howitt has a question.
Yeah, great talk, Natalie. So I'm interested in the mechanism itself. So is there a reason why some neurons maybe don't have enough synaptobrevin to actually to force activity? Because, you know, it's not an enzyme. So you're sending a synaptic vesicle protein to other neurons that are already, you know, potentially active. So is synaptobrevin rate limiting somehow in terms of activity in those snare complexes?
Yes, totally, yes. That's what I said. I was like very brief, but that's what I say, that we think it's, the effect depends on the levels of the intrinsic levels of synaptobrevin to different types of synapses. So this has been measured by a very, I think there are like only 2 or 3 papers out there that measure the levels of synaptobrevin 2 at excitatory and inhibitory synapses. And usually synaptobrevin 2 is higher at excitatory synapses. So it's kind of saturating, it's at saturating level. So I think that's why we, in wild types, we do not see any effect on excitatory neurotransmission. But synaptobrevin 2 levels at inhibitory synapses is lower. And in terms of the fusion of the vesicles, even though you need only a few copy number of the snare, so to catalyze the fusion, you only need only like 3, 4, 5 copies of the snare proteins is enough. But the more copies you have, actually, then you increase the probability of fusion of that vesicle, and you also increase the size and the duration of the fusion pore. So it also goes our results also agree with that premise that other people proposed before. And
then the donor neurons themselves, do they get exhausted then? Because, you know, they're giving up their synaptobrevin. So in terms of, you know, mechanisms, if you're giving it up, does that start to rate limit your synaptic release as well for the donors?
Well, synaptobrevin is one of the most abundant synaptic proteins. it's super abundant. Actually, if you do a knockdown, and like a classical shRNA, and you knock down 90% of the protein, you almost see no effects on neurotransmissions because it's so abundant that you need to really knock it out to see effects.
Right.
It's in excess. So I think they will not get exhausted. But interestingly, for example, there are a few works that are showing that it's one of the proteins that decreased with aging. So it's interesting.
Mm-hmm. Okay, thank you.
Thank you.
So Carolina has a question.
Yeah, thanks, Natalie. That was interesting talk, and connecting the extracellular vesicles pathway and the synaptic vesicles. That was pretty cool. And I was just wondering, because you see the correlation between the SNAP25 and CD81, whether you also do co-IP experiments and see whether the CD81 EVs really have the SNAP25. And my second question is, because you're using the pH lowering tag, have you looked into the intracellular uptake and how does that look like? Is there anything sort of like interesting coming up with that?
So for the first question, I'm assuming you mean CD81 and synaptobrevin 2.
Yeah,
Yes.
That's right. Yeah, sorry.
Yeah, that's actually an interesting question. Like, you guys probably know a lot more about CD81 than me, but it's this master organizer, right? So it interacts with a lot of protein. I have no idea if it interacts with synaptobrevin 2. Cholesterol seems to be important for all of this that we found. And cholesterol is important for synaptic vesicle fusion too. So there may be something there. Maybe, I don't know if they directly interact, but they seem to like the same type of cholesterol-rich membranes. I have no idea. I didn't do IPs. It's interesting. Yes, we want to go more in depth into that, into understanding how proteins are selected and recruited to these extracellular vesicles, how the vesicles themselves are secreted, and how they are endocytosed. We also have no idea how they are recognized and endocytosed. So, yeah, I don't know the answer, but yeah, I think we were thinking about that too. The second question was about,
I think the pH lowering tag was used to show if a vesicle is being uptake and goes into acidic compartment, they sort of fluorescence and stuff. So I'm just wondering whether you track the vesicles and in the cells.
Oh, I see.
Yeah.
No, I didn't. I want to do that, but it's actually technically hard. On one hand, the fluorine, it is going to acidic compartment, so it's not fluorescent. So to track it, we actually did some fuse to M-cherry, so it's something that will be always fluorescent, so we could track them. And the problem that we had technical was that they will be like coming in and out of the focal plane, and we couldn't really track them. This is almost, you have to think that this is almost tracking almost individual molecules, because you're tracking like coming from an extracellular, so it's technically really hard. So I thought about that. I haven't been able to do it until now, but have some ideas.
Okay. Sounds very cool. Yeah.
Just a comment before we get to Jason, you might actually connect with Bhagwan in my group, Natalie, who put M scarlet at the C terminus and has sent of CD63, not CD81, and has done some tracking. I do think that approach can be really useful for the endocytosed tracking.
Yes. Thank you. Yes, I will.
Ellie, can I just ask, so synaptic vesicles themselves are smaller than EVs typically, I believe, but would they also be purified with your purification conditions?
That's a great question. And I don't think so, because like we don't, for example, I told you synaptobrevin 2 is a very abundant protein, but the most abundant synaptic vesicle or synaptic vesicle associated protein is a protein called synapsin 1, and it's not present in the proteome. And for example, synaptobrevin 1, which is another protein integral of the synaptic vesicle membrane, is also not present on the extracellular vesicles, or we did not detect it in the proteomics. Yeah. And neurotransmitter transporters like VGLUT and VGAT, which are the transporters that load synaptic vesicles with glutamate and GABA respectively, those were also not present or not detectable in the proteomics. So we didn't detect random proteins in the proteomics, we only detect a specific set of them. So that's what I think, we do not have contamination with synaptic vesicles.
Yeah. And the difference between, CD81 and CD63 seems pretty profound in what you've shown. Have you tried to purify just straight CD81 EVs and see if all the activity is there outside of, CD63 or, CD9 or any other tetraspanners?
So actually, we did not detect CD63 in the proteomics, so I don't think these extracellular vesicles have CD63. I don't know about CD9. CD9 and CD81 work for many of the roles, they work together. So I'm actually curious to know if CD9 does the same, but right now I don't know.
Thank you.
Thank you.
Sorry, can I ask one last final question?
Yes.
So this mechanism, why do you think it's there? Why are you transferring, a synaptic vesicle protein to other neurons that already have synaptic vesicle proteins? What's the biological advantage of this transfer?
Yeah, I have no idea. I've been thinking a lot about that. I think on one hand, it could be a way of kind of, I don't know, like I can think many things. On one hand, I'm thinking like that's what I mentioned about aging. Like during aging, one of the proteins that whose levels go down with aging is synaptobrevin 2. And I'm thinking that it could be one way to kind of maintain like an extracellular pool of synaptobrevin 2 that can be uptaken into other neurons and maintain their function when their levels of synaptobrevin 2 are going down. That's one speculation. But then I said that the secretion and the incorporation was independent of activity in this short timescale. But I don't know if in the long timescale, when you have like more homeostatic changes in the network, if that could not affect the secretion, and it will be a way from one neuron to communicate the others if it's very active or very silent, and they can kind of modulate each other's levels of activity using this.
So can I ask them?
I'm more thinking of the lines of having an extracellular pool of synaptic vesicle proteins that can rescue defects on other neurons.
Yeah. Have you looked at the same system in aged neurons? As in, not from, obviously you can't get them from aged brains, but have you cultured them for longer and seen if there's a level of difference in terms of the release of synaptobrevin between a 21-day or 7-day culture or month culture?
Yeah, so this, I think I said it, for these experiments, we isolated extracellular vesicles from cultures that were around 2 weeks. 7 weeks old culture is still immature, like the neurites are still growing, synapses are not fully formed. So I don't know if I will use that, because then you will have maybe extracellular vesicles that are more involved in development too. But I could compare with cultures that are like 30 days old, for example, instead of 14.
Yep.
That's a good question. Actually, lately we've been thinking that I want to do it, but the system that we're thinking on using is actually human neurons, human induced neurons, because those live for many, many months.
Yeah.
Then you can really, and so that's the next step. We're trying first to see if we can isolate them.
Yeah.
But we're like setting the protocol first.
Fantastic.
Yeah, I just wonder if there's something that's special about EV delivered brevin, maybe it's the cholesterol you were talking about, or maybe a whole protein complex that's coming with the CD81.
Yes, I have no idea. I don't know. There is another protein, actually, I didn't say, but for trafficking on during the cycle of the synaptic vesicles, for the trafficking, especially for the endocytosis of synaptobrevin 2, it has a partner, another synaptic vesicle protein called synaptophysin. That's important for the proper trafficking. And synaptophysin, we also detect it in the extracellular vesicles. So there are hints saying that there are proteins that, you know, that with synaptobrevin 2, they co-traffic on synaptic vesicles. They're also co-trafficking to the extracellular vesicles. So there should be something like what you're saying, but I don't know.
Something cool for the future.
Yeah, but clearly, I mean, what this is also saying that is that the pathway, like the route of, you know, trafficking secretion of extracellular vesicles has some parts where it crosses with the trafficking and the cycling of the synaptic vesicles. They have some points where they talk to each other, which is interesting.
Yeah, for sure. All right, if there aren't any more questions, I don't know if you have any announcements, Carolina.
Just shortly. As you know, this month is ISEF 2021 meeting. So this is our last session before ISEF 2021. So the whole, until the end of the month, we don't have any WebEVTalk, and we will start again on June with Eva Maria Albers from Germany. So I hope everyone joining the ISEF 2021, the education day starting a week earlier. So can start joining from the 12th of May. And then the main meeting is 18 until 21 of May. That's pretty much it. So thank you so much, Natali. That was a very good project as well. I mean, I keep on thinking about, you know, how the EVs can possibly play a role because they do have the similar component with the synaptic vesicles, and your work also showing that connection. I think that was really exciting. Thanks so much for sharing it with us. And Alissa, thank you so much for leading the sessions today. Hope to see you next WebEVTalk as well.